Welcome to our first activity. Here's the breaking news. Yes, researchers have released a new high-quality collection of reference human genome sequences that captures substantially more diversity from different human populations than what was previously available. The work was led by the International Human Pangenome Reference Consortium, a group funded by the National Human Genome Research Institute, part of the National Institutes of Health. The new Pangenome reference includes genome sequences of 47 people, with the researchers pursuing the goal of increasing that number to 350 by mid-2024, with each person carrying a paired set of chromosomes. The current reference actually includes 94 distinct genome sequences, with a goal of reaching 700 distinct genome sequences by the completion of the project. The work, published in the journal Nature, is one of several papers published by consortium members. A genome is a set of DNA instructions that helps each living creature develop and function. Genome sequences differ slightly among individuals. In the case of humans, any two people's genomes are on average more than 99% identical. The small differences contribute to each person's uniqueness and can provide insights about their health, helping to diagnose disease, predict outcomes, and guide medical treatments. To understand these genomic differences, scientists create reference human genome sequences for use as a standard digital amalgamation of human genome sequences that can be used as a comparison to align, assemble, and study other human genome sequences. The original reference human genome sequence is nearly 20 years old and has been regularly updated as technology advances and researchers fix errors and discover more regions of the human genome. However, it is fundamentally limited in its representation of the diversity of the human species, as it consists of genomes from only about 20 people and most of the reference sequence is from only one person. Everyone has a unique genome, so using a single reference genome sequence for every person can lead to inequities in genomic analysis, said Adam Philip, a PhD senior investigator in the computational and statistical genomic branch in NHGRI's intramural research program and a co-author of the main study. For example, predicting a genetic disease might not work as well for someone whose genome is more different from the reference genome. The current reference human genome sequence has gaps that reflect missing information, especially in areas that were repetitive and hard to read. Recent technological advances such as long-read DNA sequencing, which reads longer stretches of DNA at a time, help researchers fill in those gaps to create the first complete human genome sequences. This complete human genome sequence, released last year as part of NIH-funded Telomere to Telomere consortium, is incorporated into the current Pangenome reference. In fact, many of the T2T researchers are also members of the Human Pangenome Reference Consortium. Using advanced computational techniques to align the various genome sequences, the researchers constructed a new human pangenome reference, with each assembly in the pangenome covering more than 99% of the expected sequence with more than 99% accuracy. It also builds upon the previous reference genome sequence, adding over 100 million new bases or letters in DNA. While the previous reference genome sequence was single and linear, the new pangenome represents many different versions of the human genome sequence at the same time. This gives researchers a wider range of options for using the pangenome in analyzing other human sequences. Beyond the report, Post Activity.